Good morning, everyone. Today, we will analyze and discuss questions from past year's exam paper. We will go through each questions in detail to understand the concepts and ensure we are well prepared for any similar questions that might appear in the future exams. Without further ado, let's get started. A student has drawn a cell structure as seen using a light microscope. The magnifications of the drawing is multiply 600. The length of the structure on the drawing is 6 mm. What is the actual length of the cell structure? To find the actual length of the cell structure, can use this formula. Actual length is equal to length in drawing divided by magnification. The length in the drawing is 6 mm, which is given in the questions, and magnifications is 600. It's also given in the question. Convert the length from mm to micrometer. 6 mm is equivalent to 6,000 micrometer. Then you have to calculate the actual length using the formula given above. 6,000 micrometer divided by 600, you get the value of 10 micrometer. So the answer will be C. The electron micrographs show part of the eukaryotic cells. Which of the labeled organelle is a site of protein synthesis? The answer is B. Inside a cell, a damaged mitochondria can be surrounded and enclosed by a membrane to form a vesicle. What happened after the fusion of a lysosome with the vesicle? After the fusion of lysosome with the vesicle containing a damaged mitochondrion, the correct outcomes is hydrolytic enzyme catalyze the breakdown of the mitochondrion. And this process is part of the autophagy, where damaged organelles are degraded and recycled. Which unit are the most appropriate to record the diameter of a lymphocyte and a red blood cells? The most appropriate units to record the diameter of both a lymphocyte and a red blood cells are D. Both types of cells are typically measured in micrometers due to their small size, approximately 10 to 20 micrometers for lymphocyte and 7 to 8 micrometers for red blood cells. Which structures will be present in a cell that causes cholera? Cholera is caused by a bacterium, so-called Vibrio cholera, which is a prokaryotic cell. The structure present in such cells include circular DNA, which is prokaryotic cell has circular chromosome, naked DNA, which refer to plasmid, which are also common in bacteria, and 70S ribosomes, typical for prokaryotic cells. Therefore, the correct answer will be 1, 2, 3. What may take place during a hydrolysis reaction? During a hydrolysis reaction, water is used to break down molecules. Specifically, a molecule of water is not produced, it is consumed. A glycosidic bond can be broken during the hydrolysis of carbohydrate. And a sucrose molecule can indeed be split into fructose and glucose through hydrolysis. And the correct answer is 2 and 3. Number 7 is a similar question which is appeared in February 2017. The question is related to the table shows some information about 4 carbohydrates and which 2 polymers form starch. Starch consists of 2 components. Amylose has alpha 1, 4 bond and is helical. Amylopectin has both alpha 1, 4 and alpha 1, 6 bond and it is branched. From the table, polymer 1 amylose is referred to helical alpha 1, 4 bond. Polymer 3 is likely amylopectin, which is branched alpha 1, 4 and alpha 1, 6 bond. And polymer 4 also likely amylopectin, which is branched alpha 1, 4 and 1, 6 bond. Therefore, The answer for these questions is 3 and 4. Which molecule is the key is sucrose? Sucrose is a non-reducing sugar and it is composed of glucose and fructose, both of which are hexose sugar. Based on the key provided, A is not sucrose, B is also not sucrose, C could potentially be sucrose, and D is also not sucrose. So the answer is C, which is the molecule that corresponded to sucrose. Which statement about triglyceride and phospholipids are correct? The correct statements are 1 and 2. 1, triglyceride and phospholipid both have a hydrophobic region, and 2, triglyceride and non-polar molecule, which phospholipids have both polar hydrophilic and non-polar hydrophobic region. Statement 3 is incorrect because phospholipid can contain both saturated and unsaturated fatty acid. When proteins are mixed with some organic solvent, hydrophobic interaction and hydrogen bonding are changed in the protein molecule. Which level of protein structure could be affected? When proteins are mixed with organic solvent, secondary structure affected due to hydrogen bonds can be disrupted. Tertiary structures is also affected is due to hydrophobic interactions and hydrogen bonds change, and quaternary structures is not significantly affected because of its focus. The focus is on single protein rather than complexes. So the best answer is A. The rate of enzyme catalyzed reaction is in human cells is regulated. What may be involved in such regulation? The answer is statement 1, 2, and 3 because all three statements are true regarding the regulation of enzyme catalyzed reaction in human cell. Changes in enzyme concentration, substrate concentration, and product inhibition all plays a role in regulating enzyme activity. The graph shows the effect of temperature and pH on enzyme activity, which statement is a correct explanation of the enzyme activity. The answer is A, where at P, hydrogen bonds are formed between enzyme and substrate. Which statement correctly describes the actions of competitive enzyme inhibitors? The correct statement that describes the actions of competitive enzyme inhibitor is they limit the formation of enzyme substrate complexes. Competitive inhibitor bind to the active site, competing with the substrate, which reduces the likelihood of substrate binding. Which rule correctly link molecule in the cell surface membrane with their roles? The correct rule that links molecule in the cell surface membrane with their role is phospholipid, which is form a barrier to polar molecule. Cholesterol stabilize the membrane. Glycolipid play a role in stabilizing membranes and cell recognition. And finally, we have glycoprotein, which function as a receptor. Actimycin is a chemical that inhibits the functions of mitochondria. Which methods of transport across the cell surface membrane would be inhibited by actimycins? The correct answer is B, because actimycin inhibit active transport, disrupt the mitochondrial function, which is essential for ATP production. And this would also indirectly affect facilitated diffusion in some cases, as the energy available for ATP may be necessary for certain transport processes. Meanwhile, endocytosis does not directly rely on mitochondrial function, so it would not be inhibited. The answer is 1 and 2 only. Which statement about the cell cycles are correct? The correct statement is all. The cell cycle consists of both interphase and mitosis. DNA replication takes place in interphase, and a cell can remain in interphase for several months, depending on the cell start. The diagram shows cells in different stages of the cell cycle. What is the last stage before cytokinesis? The answer is C. How many of the listed structures typically contain genetic material that has telomeres? The correct answer is only 2. which is Choroplast and Mitochondrion. Choroplast contain its own circular DNA with telomeres. Mitochondrion contain its own circular DNA with telomeres. And the bacteria cell does not have telomeres on its circular DNA, and while the nucleus contains linear chromosome with telomeres. What does the enzyme DNA polymerase synthesis in a cell? The correct answer is a strand of DNA using DNA as a template. Let's look a little bit about the explanation. DNA polymerase synthesize a new strand of DNA during replication and using an existing DNA strand as a template. Question number 20 is similar question appeared in October 2016. 21.2% of the bases in a molecule of DNA are cytosine. What percentage would be adenine? So to get the value, you have to look in this angle, which is in DNA, the base pairing rules state that cytosine pair with guanine and adenine pair with thymine. And if 21.2% of the bases are cytosine, then the percentage of guanine would also be 21.2%. So this account for 42.4 is actually derived from 21.2% of cytosine plus 21.2% of guanine. Then you get the value of 42.4%. Since the total percentage of all bases must equal to 100, then the remaining percentage for adenine and thymine have to minus from 100%. That means the value that you get, 42.4%, this is not the actual answer yet, but you have to get the value 100% minus 42.4%, you get the value of 47.6% for adenine and thymine combined. So because adenine and thymine pair equally, so to get the value of adenine, you have to divide by 2. Then you get the value of 28.8%. One gene provides the code for the productions of which types of molecule? The answer is D, because one gene provides the code for the productions of the polypeptide, which is a chain of amino acid that can fold into a functional protein. The table shows the mode of actions of two antibacterial drugs that can affect the synthesis of protein. If a bacteria are treated with both drugs, what will be the immediate effect? So the answer is statement 1 and 2, which is transcription will stop, but faulty protein may continue to be synthesized. And statement 2 is correct. If translation has started, protein may be faulty. The diagram shows transverse sections of part of a plant. This is also a similar question appeared in February 2018. And in the transverse section, which tissue transports sucrose? The answer is C. And sucrose synthesized in green leaf is transported via the folium. Which statement describes movement through a plant in the apoplast pathway? So the correct statement is A. Water moves through the cell's wall. In the apoplast pathway, water travels through the extracellular spaces and cell wall by passing the cytoplasm, which changes to the water potential and the volume of liquid in the folium occur when sucrose is moved from the folium sieve tube to an actively dividing root tip. When sucrose is moved from the folium sieve tube to an actively dividing root tip, water potential in folium sieve tube become more negative as sucrose concentration increase in the root tip, drawing water out of the folium. And the volume of liquid in folium sieve tube decreases due to the water leaving the folium to follow the sucrose. So the correct answer is C. Water potentials in folium sieve tube become more negative, and the volume of liquid in folium sieve tube decreases. The diagram shows the result of an experiment using leaf with the same surface area from two different species of plant. Each leaf was left on a balance in daylight in a closed room, and it mass recorded at 1 hour interval. Which features of leaf 2 could explain this result? So, the answer is statement 1, more stomata per unit area of leaf, fewer trichomes hair of the leaf, and thinner cuticle. Which evidence support the cohesion tension theory for the movement of water in flowering plants? The answer is statement 1 and 3. Cardiac muscles is made up of many fibers that form the wall of the chambers of the heart. When the heart contracts, these fibers shorten in length, so the muscles create a force that exerts the pressure on the blood, causing it to move. Which statement explains the difference in thickness of the wall of ventricle of the heart? The answer is the number of muscle fiber in the left ventricle is greater than the number in the right ventricle, so their contractions has more force, exerting more pressure on blood. Which reaction takes place at a higher rate in a capillary in an alveolus than in a capillary in active muscle? This is also a similar question, appear in May 2022. In the alveoli, gas exchange happen, leading to higher rate of reactions that convert bicarbonate back to the carbon dioxide. In active muscles, more carbon dioxide is produced, favoring reactions that involve carbonic acid and hemoglobin binding with carbon dioxide. The reactions that occur more in the alveoli is hydrogen carbonate ion plus hydrogen ion to produce carbon dioxide and water. The diagram shows the effect of 3 different concentrations of carbon dioxide on oxygen dissociation curve for human hemoglobin. And what effect does increasing carbon dioxide concentration have on hemoglobin? This is also a similar question appeared in October 2020. So the answer, it makes it more efficient at taking up oxygen and less efficient at releasing it. The graph shows the changes that take place in the volume of the left ventricle during one cardiac cycle. Which point of the graph represents the start of atrial systole? The answer is C. The following tissues carry an electrical impulses during the cardiac cycle. And in what order does the electrical impulses travel during the cardiac cycle? The answer is 4, 2, 4, 2, 1, and 3. This sequence represents the pathway starting from the sinoatrial node, then through the muscle wall of the atria, to the atrioventricular node, and finally to the Purkinje tissue. Some of the effects of smoking are listed. Which component of tobacco smoke cause this effect? The answer is A. Coffee can cause by tar, increased blood pressure is caused by nicotine, decreased oxygen transport is caused by carbon monoxide, increased risk of cancer caused by tar, and prevent cilia from moving is also caused by tar. So the best match is A. The photomicrograph shows a section through part of a bronchus wall. What is the functions of the tissue label? The functions of the tissue label X is support the airway to prevent collapse. Which of the following increase the risk of contracting TB? The correct answer is B, which is statement 1, drinking unpastured milk and living in overcrowded condition. Drinking unpastured milk can increase the risk of contracting tuberculosis, specifically involving TB, and living in overcrowded condition increase the risk of transmission of TB in general. So eating shellfish that have fat on raw sewage is not directly linked to TB risk. One control method to reduce the spread of malaria is to use insecticide. It can be used to treat mosquito net. Another control method is to completely cover area of water with insoluble polystyrene ball that float on the surface. Using this information, there are the reason for this control method, which is the answer is net treated with insecticide, which will kill some mosquito, and the polystyrene ball restrict the breathing of the mosquito larva. What is not an example of antibiotic action? The correct answer is D, stimulation of antibody production. This is not an example of antibiotic action, but antibiotic typically work by damaging cells membrane, preventing protein synthesis or inhibiting cells wall synthesis. Which row correctly identify the role of B lymphocytes and T lymphocyte? B lymphocytes are responsible for antibody production and the humoral response, while T lymphocyte primarily secrete cytosine. A person's blood group is determined by antigen present on the red blood cells. The table shows the antigen and antibodies in the blood of people with different blood group. This is also a similar question which is appear in May 2022. During the blood transfusion, it is essential that the recipient blood does not contain antibody to the donor blood. Which blood groups can be given to a person with blood group B? The answer is none from the listed. The answer is A, B, AB, and O. A person with blood group AB has both A and B antigen on their red blood cells and does not have antibody to A or B in their plasma. This means that they can receive blood from individuals with any blood type as their immune system will not react against the A or B antigen present in the donor blood. An enzyme hydrolyzes the too heavy polypeptide change of an antibody molecule. The hydrolysis occurs at the hinge region and break the antibody into 3 fragments. How many of these fragments are able to bind to antigen? The answer is 2. When an antibody is hydrolyzed at the hinge region, it typically results in 2 fragmentation, which can bind to antigens, and 1 Fc fragment, which does not bind to antigen. That concludes today's sessions. I look forward to seeing you at the next session, and please subscribe to Jom Stabila YouTube channel. Don't forget to turn on your notifications so you don't miss any updates. And best of luck in your studies, and wishing you every success. Thank you for your support and cooperations.